There's a stretch of DNA that stayed completely identical in chickens, rats, crocodiles, and chimpanzees for over 500 million years. 500 million years of evolution, mass extinctions, continental drift, 5 ice ages. And that stretch didn't change a single letter in any of those species. Then came the human lineage. And in less than 7 million years, that same stretch changed 13 times. Only in you. The researchers who discovered this gave these stretches a special name, human accelerated regions. And HACNS1, the one that changed 13 times, has the strongest acceleration signature of any sequence in the entire human genome. What does this 81 base pair stretch do? The current hypothesis is that it shaped the development of the wrist and thumb. The opposable thumb, the piece that made tools, writing, surgery, and music possible. Right now, you're here, watching this, holding a phone or a coffee cup with a thumb that can swing toward your palm in a way no other primate can replicate with the same precision. And part of the reason may come down to those 13 changes in a stretch of DNA that the rest of the vertebrate world never touched. But the thumb is just the beginning. Because if you look at the full set of changes that separated your lineage from chimpanzees over the last 6 to 7 million years, what emerges isn't a list of different genes. It's something far stranger and far more fascinating than that. Today, you're going to understand exactly what's written in your genome that isn't written in any other living animals, and why the answer will change the way you think about what you are. Let's start with a number that looks simple, but hides something unsettling. The DNA sequence difference between you and a chimpanzee is approximately 1.23% in individual nucleotide substitutions, with additional deletions and insertions covering roughly 3% of the total genome. Now compare that with this. Birds and reptiles diverged 310 million years ago. 310 million. And yet, a lizard and a pigeon share more visible morphological features with each other than you and a chimpanzee share in terms of cognitive capacity. Think carefully about what that means. The separation between humans and chimpanzees is 50 times more recent than the separation between birds and reptiles. But the cognitive distance between you and a chimp is incomparably greater than anything separating a lizard from a seagull. Language, mathematics, philosophy, art, medicine, architecture, the ability to ask where you came from. None of that exists in any other lineage, and all of it emerged in a time frame that, on a geological scale, is practically a blink. That raises a question evolutionary biologists take very seriously. What happened in this lineage that was so accelerated, so disproportionate to the time available? And the answer starts with a shift in how we think about what genes actually do. Because most people, when they hear genetic difference, picture slightly modified versions of the same parts. A slightly different gene produces a slightly different result. Intuitive, reasonable, and completely wrong as a model for understanding what happened here. Genes don't work like individual recipes where one different ingredient changes one flavor. Genes work in regulatory networks, where a single gene can control the expression of dozens of others, and where one change at a specific point can completely reorganize the developmental pattern of an entire organ. Think of an orchestra. You can swap one violinist for a slightly different one, and the result changes a little. But if you change the conductor, or change the score the conductor is following, the same orchestra with the same musicians produces an entirely different symphony. The difference between you and a chimpanzee isn't mainly about which proteins you produce. It's about when, where, for how long, and in what amounts those proteins are produced during development. It's a question of regulation, not composition. And here's the part that changes everything. That means a single change in the right place, not in a gene that makes a protein, but in a stretch of DNA that controls when other genes are switched on, can have effects that ripple through entire developmental cascades. researchers identified specific stretches of the human genome where this kind of regulatory change occurred at extraordinary speed. Stretches that stayed frozen, identical, for hundreds of millions of years across all vertebrates, and that suddenly, in the human lineage, started accumulating changes at rates far above what random mutation would produce. The statistical signal is unambiguous. This is an accident. It's intense positive selection. Mutations that were strongly favored because they conferred real advantages in survival and reproduction. Something was pushing these changes forward. And what was being built wasn't a new protein. It was a new instruction for how to use the proteins that already existed. But which instructions specifically and what each one did is where the story gets really strange. If you're finding this fascinating, the idea that what makes you human might not be in your genes themselves, but in the instructions for how to use them, subscribe and hit the bell. Every week we go this deep. Don't miss the next one. So back to HACNS1, because now that you understand that what matters is regulation, when and where genes are switched on, the story of this 81 base pair stretch becomes much more impressive than it seemed in the opening. HACNS1 is not a gene. It doesn't make any protein. It's an enhancer, a stretch of DNA whose only job is to control when and where neighboring genes are activated during embryonic development. And this particular stretch stayed so conserved for so long because it was clearly doing something important. Vertebrates that altered it likely didn't survive to pass it on. 500 million years of evolutionary pressure keeping that stretch untouched across virtually every lineage with a backbone. Then researchers took the human version of HACNS1 and the chimpanzee version and placed them into mouse embryos to see what each one did. The chimpanzee version activated genes moderately in the forelimbs. The human version lit up a specific region, the anterior wrist and the base of the thumb. 13 substitutions in an 81 base pair window. 13 changes that, accumulated in the lineage that led to you, appear to have reprogrammed forelimb development in a human-specific way, potentially contributing to the hand morphology that makes fine manipulation possible in a way no other primate can match. The opposable thumb isn't just convenient. It's the interface between mind and world. Without it, no flint tools, no needle, no paintbrush, no writing, no surgery. The chain connecting those 13 DNA changes to the James Webb Space Telescope is indirect, but real. And while HACNS1 was reshaping the hand, another stretch of the genome was doing something even more radical to the organ that would use that hand. If HACNS1 has the strongest acceleration signature among all non-coding enhancers, HAR1 has the strongest acceleration signature across the entire human genome, full stop. And what makes HAR1 so fascinating is what it actually is, not a gene that makes any protein. CHAR1 is an RNA gene. It produces an RNA molecule that acts directly without being translated into protein, an instruction that speaks directly to the cellular machinery. This RNA molecule, called HAAR1F, is active in the developing human brain between the 7th and 19th gestational weeks. 7th to 19th week. You didn't yet have a recognizable human form when HAAR1F was already at work in your cortex. It's expressed specifically in what are called Cajal retzius cells, a specialized type of neuron that acts as a coordinator of cortical development, guiding other neurons into correct positions as the neocortex layers are being built. To understand what's at stake here, the neocortex is the outer layer of the brain responsible for reasoning, language, planning, and creativity, everything you consider uniquely human. And HAAR1F is active exactly during the window when those cortical layers are being assembled, neuron by neuron. The chimpanzee version of HAAR1 differs from the human version at 18 positions. 18 changes that appear to have altered the three-dimensional folding of the RNA molecule, because the human version adopts a different spatial structure than the chimpanzee version. Different structure, different function. different brain. But building a bigger brain isn't enough on its own. Neanderthals had larger brain volumes than ours, and yet something in the internal organization of the sapiens' brain produced results that Neanderthals didn't replicate in the same way. Which brings us to a gene researchers call, with some exaggeration but not without reason, the language gene. FOXP2 is a transcription factor, a protein that regulates dozens of other genes, and is among the most conserved proteins across all mammals. So conserved that it barely changed across tens of millions of years of mammalian evolution. But since humans diverged from chimpanzees, FOXP2 accumulated 2 additional amino acid changes at positions that had remained essentially identical across mammalian history. The statistical signal of positive selection is strong. Something was favoring these variants with force. When researchers introduced the human versions of these changes into mice, the animals vocalized differently, producing more complex sounds with better motor control of vocalizations. FOXP2 isn't the language gene in any simple sense, but it's clearly an important component of the cognitive and motor apparatus that makes human language possible. Now here's the part nobody expected. When researchers sequenced the Neanderthal genome and looked at FOXP2, they found exactly the same 2 human changes. Identical. Neanderthals had the human version of FOXP2. That means these changes occurred before the split between Homo sapiens and Neanderthals, estimated at between 300,000 and 400,000 years ago. The genetic apparatus that contributes to human speech is older than our own species. Neanderthals and sapiens shared this piece of the puzzle, and yet something differentiated the cognitive outcomes of the 2 lineages. What that tells us is both unsettling and fascinating. FOXP2 alone doesn't explain human language. It's one piece of a much larger system, and the other pieces were being assembled in parallel in other parts of the genome over millions of years. And one of the most recent and most surprising pieces of that system was found somewhere. Nobody was looking. Remember the hook? That stretch of DNA conserved for 500 million years changed 13 times only in humans? The logic that a regulatory change in the right place can have enormous downstream effects. Notch2NL is the most dramatic example of how that principle plays out, and of how evolution rarely offers advantage without cost. Notch2NL arose from a gene duplication. At some point in the evolutionary history of the human lineage, an older gene, Notch2, was duplicated incompletely, producing a broken copy. That broken copy sat in the genome long enough to accumulate additional mutations that, by accident or selection, made it functional in a new way. This new human-specific gene, which doesn't exist in chimpanzees, gorillas, or any other non-human primate, acts on cortical progenitor cells in the fetal brain. It delays differentiation of these cells, keeping them in division mode longer. More divisions, more neurons, larger cortex. When researchers introduced Notch2NL into mouse brain organoids in the lab, small structures that mimic fetal brain development, the organoids produced more neurons, and for longer than controls without the gene. The hypothesis is direct. Notch2NL was one of the triggers behind the extraordinary expansion of the human neocortex. Around 86 billion neurons, an estimated 100 trillion synaptic connections. The most complex object we know of in the universe, and part of the recipe that built it may have started with a broken copy of a gene that fixed itself in exactly the wrong right way. But here's the cost. Notch2NL sits in a region of chromosome 1, called 1q21.1, that became unstable precisely because of the duplications that created it. When extra copies of Notch2NL appear in that region, the result tends toward macrocephaly and increased risk of autism. When copies are deleted, the result tends toward microcephaly and increased risk of schizophrenia. The same locus that expanded the human neocortex created a point of genetic fragility that didn't exist before. Evolution isn't clean engineering. And what comes next will show you that the greatest trick of our lineage wasn't any specific gene, but something far more surprising. A chimpanzee reaches sexual maturity at 8 or 9 years old. A human takes between 13 and 17. That's widely known. What isn't widely known is what happens at the genomic level during that extra window, and why it may be the single most transformative factor in the entire human story. In 2009, researchers at the Max Planck Institute published a study in PNAS analyzing gene expression in the prefrontal cortex of humans, chimpanzees, and rhesus macaques across development. What they found was unexpected. The human brain isn't just bigger than the chimpanzees. It's genetically slower. The transcriptional development of the human prefrontal cortex, the region responsible for reasoning, planning, impulse control, and complex decision-making, is delayed relative to the chimpanzees. Not uniformly, but in a specific subset of genes linked to neural development. The human brain matures more slowly by design. This phenomenon has a name: neoteny. It's the retention of juvenile characteristics into adulthood. And in the human lineage, it shows up not just in morphology, but in the actual gene expression patterns of the brain. Adult humans retain patterns of cerebral genetic activity that in chimpanzees are characteristic of juveniles. And the implication of that is profound. The window of maximum neuroplasticity, the period when the brain is most open to learning through experience, when new connections form most readily, that window in humans stays open for years longer than in any other primate. Think about what that means concretely. A young chimpanzee learns what it needs to know relatively quickly, and the brain closes. A human spends years and years in maximum absorption mode, being shaped by environment, culture, language, and the people around them. The long childhood isn't an evolutionary vulnerability. It's the programming mechanism of the most sophisticated computer nature ever built. And it wasn't an accident. The genes that control the pace of development, when the body stops growing, when the brain closes its plasticity windows, when sexual maturity is reached, show clear signs of having been selected differently in the human lineage. Something was choosing, generation after generation, the individuals who took longer to grow up. Because taking longer to grow up meant learning more before closing. And learning more meant surviving better in an environment where the primary survival resource wasn't physical strength. It was accumulated cultural information passed between generations. Neoteny doesn't just explain the long childhood, it connects every other block in this story. A larger brain, built by genes like Notch, 2NL, and HAR1, only becomes fully advantageous if there's enough time to program it. And that time, that deliberate extension of the learning window, may have been the multiplier that turned all the other genetic changes from modest advantages into something qualitative and unprecedented in the history of life on Earth. But before we get to the conclusion, we need to talk about something the story of human evolution tends to leave out, and that completely changes the way you'll understand what this story means. Every advantage we described today came with a cost. Notch 2NL expanded the neocortex and created a chromosomal instability point that didn't exist before. The long childhood maximized plasticity and also created decades of dependency. Human children are the most vulnerable and most care-intensive of any mammal on the planet. The human brain consumes roughly 20% of the body's total energy at rest, despite representing only about 2% of body mass. No other mammal pays that disproportionate a metabolic cost. The most powerful brain on the planet is also the most expensive to run, and that created pressures around food, social cooperation, and the need to cook food to extract more calories that shaped the entire trajectory of the species. Evolution isn't an engineer designing clean solutions. It's a process that favors what works now, with the available materials paying whatever costs the environment will tolerate. What that means is there's no perfect version of human waiting at the end of a linear evolutionary trajectory. What exists is a set of instructions that has been adjusted, patched, duplicated, and repurposed over millions of years, and is still being adjusted right now. And here's the thread that connects everything you saw today back to the beginning. Remember the question from the hook? Why did 1.23% of genetic difference produce a cognitive distance that has no parallel in any other species divergence in the fossil record? The answer isn't that humans have magical genes other animals don't have. The answer is that the human lineage accumulated changes at the regulatory level, in the when, the where, the how much, in such a way that the result was qualitative, not just quantitative. We are not chimpanzees with different parts. We are the outcome of a different set of instructions applied to nearly identical raw material. And that has an implication that goes beyond biology. If what separates you from a chimpanzee isn't in the ingredients, but in how those ingredients were organized over time, then what you are isn't fully defined by what you have. It's defined by how what you have gets expressed, by the when, by the how much, by the where. That logic holds for genes, and possibly for far more than genes. So here's what sticks with me after going through all of this. You are the product of 6 to 7 million years of accumulated regulatory adjustments. Stretches of DNA frozen for 500 million years that started moving in your lineage at a pace that had no precedent. Duplicated genes that broke and fixed themselves in exactly the wrong way. An absurdly long childhood that your genome chose, generation after generation, because learning more was worth the cost of closing later. All of that resulted in an animal that burns 20% of its own energy just to maintain the organ that is processing this information right now, and uses that organ to ask why it exists. No other species we know of comes close. And the deeper science goes into that question, the more the answer seems to be not we're special because we have something unique, but we're special because of how we reorganized what everyone else already had. The difference between you and a chimpanzee isn't a difference in ingredients. It's a difference in the instructions. The same basic components arranged differently for long enough produce things so distinct that the common origin is hard to recognize. Until you look at the genome and see it written there in every cell of your body. What strikes you as most surprising about all of this? That the difference is so small in terms of raw material, or that it's so enormous in terms of outcome? Because those are two very different ways of looking at what you are. And if the logic of when and how much and where applies to genes, if what makes you human isn't what you have, but how what you have gets expressed over time, what does that say about potential? About what can still be activated, amplified, reorganized? Evolution hasn't stopped. Your genome isn't a closed archive. It's a living system, still being shaped by pressures we're only beginning to identify. What the story of the human accelerated regions tells us is that the greatest transformations don't always come from new ingredients. Sometimes they come from a different instruction for how to use what was already there. If you enjoyed this story, subscribe. There are plenty more where this came from. See you in the next one.